Today we're digging into a huge challenge, cancer. The search for better treatments is always on, and we're looking at some innovative research that's trying to find new ways to attack cancer. And what's very interesting here is how this research gets right down to the molecular level. We're talking about protein interactions, or PPIs, and these really critical spots on them called binding pockets. The authors have created a large comprehensive 3D map of what they call ligandable protein pockets across different cancers. Ligandable, meaning places drugs can actually bind, and this is in a way shifting how we think about drug discovery. It's potentially unlocking targets that people used to call undruggable. Maybe we step back for a second. Why are these interactions so important to target anyway? Well, fundamentally, PPIs are how cells get things done. They're essential. But when they go wrong, when they get dysregulated, that's often what drives cancer growth and progression. Then PPIs become part of the problem. So stopping those faulty interactions is a key strategy. But how do you find and map these pockets? What did the authors do? It was a huge undertaking. They built a detailed 3D data set, over 300 cancer-related PPIs studied across 12 different types of cancer. They looked at what they call OncoPPI targets, which are PPI uniquely activated in tumors versus healthy tissue. First, they start mapping pockets, and what did they find? Are all these pockets the same, or are there different kinds? Well, definitely different kinds. They found three main types. First, you've got interface pockets, right where the proteins meet. They're located directly at the interaction site. Finding one of these gives you a direct way to just block that specific protein pairing and disrupts that particular pathway. The second type involves allosteric pockets. These are not directly at the interface. Binding there can change the protein's shape or function in a way that still affects the interaction. These are really interesting for designing small molecules, including PROTACs. That is, molecules that help degrade unwanted proteins. How does that work? A PROTAC molecule basically has two ends. One grabs onto the target protein you want to get rid of, and the other end grabs onto the cell's own machinery for protein disposal, like the ubiquitin proteasome system. PROTAC brings them together, so it flags the bad protein for destruction. So far, we have direct interface pockets and indirect allosteric ones. What is the third type? Equilibrium pockets. They are unique because they only form when the two proteins actually come together and interact. The pockets are not really present on the proteins when they are separate. It means we can find druggable sites even on interfaces that look completely flat. Thus, surfaces or proteins that in the past would have been considered as fully undruggable using small molecules. Now, is this still theoretical, or is there immediate potential? Well, it is not just theoretical. The authors found that many of these newly mapped pockets can actually bind known ligands already, over 600 unique compounds, and 17 of those are already FDA-approved drugs used for other things. This shows that some of these pockets are genuinely druggable, and this study also highlighted hub proteins. That is, the most connected ones in the cancer networks. Targeting a hub protein could potentially disrupt multiple cancer-driving pathways all at once instead of just one. They found over 100 hub proteins. The authors also noted that some cancers exhibit a higher number of hub proteins, while others are dominated by proteins with fewer interactions. Several novel pockets are discussed in the article, and this whole 3D map is available for other researchers. So just to wrap up, this detailed 3D mapping isn't just finding more targets. It refines our understanding about cancer and provides concrete starting points. The study also makes you think if this kind of analysis helps finding and prioritizing targets in cancer, what if we could apply the same approach across all kinds of diseases? Could this change the game for therapeutic development way beyond cancer? That's it for today. Thanks for tuning in. Until next time, take care.